Welcome back to another episode of Concentrating on Chromatography. Ansgar, thanks so much for joining me today.
Yeah, thanks, David, for the invitation. Looking forward to this episode.
So, mzio grew out of the open source mzmine community. Can you walk us through how that project evolved into a commercial software company and what made you decide that this was the right time to build a business around the platform?
So basically, mzio has its roots in academia, and it all started with a PhD project. Actually, it was in Finland in 2004. And it was one PhD student in the lab of Matteo Resic. And it was the time around when high resolution mass spectrometry really kicked off with the arrival of the Orbitrap. Time of flight instruments got better. And the machines, they produced more and more data. And like in other occasions, instrumentation was faster than software on the development side. So scientists had to build their own stuff. And this is basically what the project was all about back in the day. And they've published mzmine 1 in 2006. And quickly after that, our co-founder, Tomasz Puskal, joined the project. And in the very beginning, Matej had the idea what was ever built in his lab should be open and accessible for others so others can build on. And Thomas basically took over the project and he created mzmine 2, which he published in 2010. And that really is an important step because with his software engineer background, he really transformed how the project worked. He created a modular framework. So basically, he coded the core of mzmine in a way that no one needed to touch it and others could add processing modules. So it was very easy for other scientists to add modules without breaking the rest. And this made it possible to create a real open source project because now we could put it on GitHub and others started to join in. And this is basically also the story how myself and the other co-founders joined the project because I started my PhD in analytical chemistry in 2017. And I had the same issue. I was looking back then into Orbitrap data from a Q-exactive machine, mainly lipidomics, and I needed a good lipid software for lipid identification. And there was clearly a bottleneck also in that regard. So I thought, okay, there's something important to do here. And I thought, let's start from scratch. And I quickly realized, no, that doesn't make any sense. And luckily, together with Robin Schmidt at the time, the now CSO of mzio, he was at the same university as me, University of Münster in Germany. And he discovered the mzmine project. And together we jumped on it and started to add modules. And we approached Tomasz at a certain point in time and told him, hey, look, we are looking into your software. Is it okay if we contribute? And he was like, yeah, of course. This is what it's all about. Yeah, and long story short, a few years later, Steffen Heukerud joined. Together, we rewrote basically the whole thing again, modernized it. I mean, 2010, quite a long time, a lot of technology changes also happening in computer science. During that time, we went different directions. So at the time, I have joined instrument vendor. I developed their software solutions, later worked as a product manager. And Robin went the academic route. At that point in time, he was a postdoc in Peter Dorstein's lab in San Diego. And then it was 2023. Steffen, who joined us a bit later, just finished his, or was about to finish his PhD. And we thought about how to continue this because we really wanted to keep mzmine alive. And the challenge here is always, if you have a project that lives in academia, it basically requires funding from various sources, however you get money, and also on individuals that come after you that are willing to continue working in this project. And this has worked 2 times for mzmine. So we were 2 times lucky, but you never know. And we clearly saw the need also in the field. And we already had a huge user base globally at that point in time. So we decided, okay, let's try. Let's found a company around this and see how we can further improve it and also to maintain it.
And one thing my listeners will be really excited to hear is that you've very much taken a vendor agnostic approach to positioning in a market where a lot of instrument vendors have their own data software, obviously, and it's part of a larger ecosystem. How do you make the case to labs for adopting a platform that sits outside of that vendor stack?
Yeah, that's a good point, and this is basically where we wanted to tackle the next challenge with the new generation of mzmine, with mzmine 4 and mzmine Pro, because before we had a strong focus on liquid chromatography and MSMS data from high resolution instruments. But when you look at a modern lab, independent of the industry, could be biotech, pharma, but also could be food, there's usually they don't rely on a single vendor. Usually they don't rely on a single platform or technique. So you will always find GC instruments. You will also find LCMS instruments. And also you will find different MS detectors. Because I think at this point in time, there is no clear gold standard. We have at least 6 major vendors and even more MS vendors. It's a highly competitive field. And all instrument vendors make a great job in delivering on different strengths. So I don't see it coming very soon that a lab will solely rely on a single vendor. So basically, everyone has that challenge to handle multiple results data files. And this comes with a big overhead, having multiple softwares. These softwares are usually differently maintained. Some vendors do a great job in maintaining their software. Others, yeah, sometimes you rely on a solution that is already discontinued, depending on your data analysis needs. And if you are only having to use one platform, I mean, it's really nice from the user perspective. You, you need less training. If you learn the software once, you get a new instrument. Data analysis is not really a fear you still have. I think this is also something we hear a lot. And also, when I was working for a vendor, if you have a customer or a user who's used to a certain platform, some may be reluctant to do a change. I think the only real reason to make a change if the other technology is really disruptive. But if it's only a marginal improvement, you may just stick with what you know. And if the analysis software is already is solved and it doesn't make a difference, I think that's a huge step also for any lab to be more brave to go for other MS solutions as well.
And that's a great transition because one thing that really stuck out to me when I was reviewing the platform was how remarkably broad the scope is. And on the podcast, we've spent a lot of time focused on a lot of projects that focus primarily on LCMS and GCMS, but I've also started to learn more about ion mobility and other topics. How do you prioritize development across the different modalities without spreading yourselves too thin?
Yeah, this is always debate, but usually it's quite easy because it comes from the users. And with mzmine, we have this large user base, and we try to create a platform or a forum where people can always bring the latest questions, trends, and technologies to us. This can be done on our, for example, GitHub page, but also just reaching out directly to us at a conference or via email. I know we also have customers, obviously. They also, of course, have their strong desires for certain technologies. But it really comes to, there's no clear prioritization. It really comes to what most people want. And also on the technology side of things, we try to leverage this module platform because a lot of these algorithms we have developed for LC can be reused for GC data, for example. Right? You only need one module to build chromatograms. Chromatograms are always the same. Resolving peaks is always the same. you may need different parameters compared to when you compare GC or LC. But the whole idea is don't develop everything again from scratch. Use what you have and add the missing pieces. For example, for GC, when we started with GC, yeah, obviously you have electron ionization, hard ionization, you need spectral deconvolution. So we just created a module for that and created then a workflow and decided, okay, this has to sit here in the workflow. Now let's give it to the community and let's collect feedback. And this is basically how it works for us. And so far, yeah, we get lots of good feedback and even more work to do, to be honest, because the field doesn't stop.
Yeah, new technology and new data sources for sure. One thing I was very impressed reading about was the Fair MS initiative and how you're making historical MS data searchable and reusable for labs. Could you touch on that initiative?
The Fair MS project or initiative is now a new funded project. We are very certain to get funding from the city state of Bremen. I think most companies and also scientific groups, they sit on a gold mine when it comes to knowledge and data. But this data usually sits in folders on your hard drive. And it's very hard to readdress a project that is older than maybe 5 years because in academia, maybe the PhD or postdoc has left, or in a company, the individual scientist who worked on the project has moved on, or in another position, or maybe in another company. Let's say you start a new project, which is maybe related to the old project. How will you be able to compare these data sets on top of the human that is maybe or has moved on? Maybe technology has also changed. So maybe the new software you have for your new instrument doesn't work anymore well for the old technology. This is often the case if they're like 10 years in between. So I think it is then very important to build something that can actually access all these different types of data and is able to compare it. And we've been leveraging here modern AI technologies to transform the raw data into something readable and comparable across all these platforms. mzmine here is, let's say, the hub we can use as a processing tool because it already starts when you try to compare different raw data with the readability of the raw data. So not every tool or can read any mass spec vendor raw type. And let's say we use mzmine as a processing pipeline, and we can then use the mzmine results for the FairMS initiative project. And on top of that, we can then leverage open source deep learning models. And there are great examples currently in the field. To name a few, we have MS2 Deep Score, and we also have Dreams. And these models, they are trained on MS2 spectra. And with these models, we are able to calculate embeddings of our results. And these embeddings make it then possible, not just in terms of speed, but also, yeah, with these embeddings, you are able to compare across all these different results coming out of mzmine from all these different projects.
And I believe you had mentioned Bremen, Germany in response to the funding. The area certainly has a rich history of mass spectrometry. How has being embedded in that scientific community really shaped the product philosophy?
Yeah, it's really cool to be in Bremen because we have Bruker Daltonics and Thermo Fisher Scientific right around the corner. And Bremen is not a huge city. It's really nice to, yeah, you have all these people around you. You can always go to the vendors and speak to them directly, but also in your everyday life, you have interactions and you obviously meet on private occasions. So it's really great to have these scientific connections in your everyday life.
I'd love to spend some time on the 2026 release of mzmine and the improvements that have been made in terms of the new lipid and small molecule dashboards and an impurity analysis workflow. Can you talk a little bit or provide an example of how these tools can really impact a chemist's day to day workflow?
So the main goal is to make the final results even more easy to interpret for the scientist. So our goal is to give out high quality results so that the scientist can focus on the actual science. And with mzmine, we have like a long history in being very modular and flexible, but also that there's like a steep learning curve. And we try to now leverage all these functionality we already had in the past and make it all even easier. And now with this initiative that we started to implement more and more dashboards, we really just try to directly give the user the information that he really needs. My personal favorite, I have to admit, I did my PhD in lipidomics. The cool thing with lipids is everything is the same. That makes it sometimes easy to analyze, but also very, very hard because some of these MS2 spectra, they basically look identical. And I have learned in the past 10 years, you have to really leverage all the information you can get from your instrument. MS1 alone is not enough. MS2 alone is not enough. Retention time alone is not enough. So you have to really combine everything. And with this new dashboard, we really tried that and also included a new score that also includes all these different types of information, cross-reference against other annotations, but again, making it easily visible into one single dashboard so people can just click through all the annotated lipids and make up their minds. Is it a real lipid or is it a false positive or a false negative?
Now, this probably doesn't surprise you, but of the 65 or so episodes I've recorded so far over the last year and a half, a lot of them end up touching on AI in one way or the other. You've described Fair MS as AI powered. What role is AI actually playing? Is it more machine learning for spectral matching or deep learning for annotation?
Yeah, it's more the transformation of the data into embeddings to enable the matching and also the speed. It's a very easy,
No,
short
that's
answer.
great. No, that makes a lot of sense. And I could certainly see how, just as a total novice, I would think that with all the data sources you're compiling, just that unseen pattern matching that people struggle with, that AI has proven successful with in terms of like the protein folding project years ago. A great example of some of that pattern recognition that we just can't do and the machines can do a little better than us.
I think this is a great example where AI is really outstanding and finding patterns. I mean, there's so much debate what AI can do and what AI may replace. I think, especially in mass spec, we are definitely still in a phase where AI to help us to develop new fascinating tools, but it will not yet replace a scientist, right? It will always be with people in the loop. AI is certainly very helpful to guide decision making, but I think we are very far away from getting automated decisions from an AI, we should rely on.
Oh, I totally agree. Would you be able to introduce the LIMMIC project? I understand it tackles uh, calibration simultaneously across a lot of different factors, but I'd love to hear a better explanation from you.
Another project where we were fortunate to get funding from the European Union. And this is a joint project with the CMOS group in Mannheim from the group of Carsten Hopf and also a Swedish company called Polymer Factory. And here we bring together us as a software company, Polymer Factory as a provider of chemicals, polymers for calibration, and also application specialists in the CMOS group. Hop's team, they do also a lot of imaging techniques, spatially resolved analysis, but usually also in combination with, for example, ion mobility and also orthogonal information from LC-MS. And the main challenge, I think, in calibration is to find chemical standards that are on one hand stable in all these different conditions, but that also work equally well in all these different techniques. For example, something that works very well with electrospray to calibrate masses doesn't necessarily be a good standard for ion mobility. And yeah, what the guys at Polymer Factory have is a very interesting standard mixture already that works really well across all these techniques. And now we are working together to extend this product they have to even lower mass ranges. So currently we cut off usually at 300 Dalton, but for metabolomics we want to go lower as well. And our task also is to make this again like more of an automated process, so that then, for example, the scientist in Carsten Hopf's team can simply rely on the software and the automatic calibration across all these different modalities.
And one thing that stands out is how wide your application list really is. I mean, you're spanning drug discovery, lipidomics, metabolomics, polymer chemistry. Where are you seeing the fastest adoption right now? Obviously, we're both just getting back from ASMS, so I'm sure you got a fresh round of feedback, but where's most of the excitement regarding the platform?
So first of all, I think we span such a wide range of applications because the analytical techniques behind it are very similar. And also the challenges are very similar. If you are in the field of small molecules, it doesn't really matter if it's metabolites or if it's, I don't know, pollutants, if you're in environmental science. So the MS method, the application setup remains very, very similar. And also the software data processing. It's, for us, it's really broad where we see the adoption. We obviously started mostly in pharma and biotech related markets, but we see a huge adoption also in food and applied markets for us. So that was really interesting to see. And for me, it was also very interesting to find out which companies are coming to us where I wasn't aware that they're running their own mass spectrometry instrumentations, but also in the environmental science. I think, especially with tracing of PFAS and applications like that, which becomes more and more mandatory from a regulatory perspective. Also, public sector government organizations are approaching us if we can help them with establishing a solution. So, for me, no clear winner in terms of what is growing the most. Just a general increase of need regarding software solutions to handle the vast amount of data.
Well, in a sense, that's very, very good news and very promising. PFAS analysis is a area I wanted to discuss because it is such a popular regulatory priority right now. I believe in April, Japan just installed their newest round of regulations, and I know the EU had installed some for this year. With methods like EPA 533 and 537.1 driving lab investment, how does your platform really fit into that non targeted workflow?
Yeah, again, I mean, these methods, they mainly have their roots in metabolomics screening, these non-target screening, LCMS, MS, high-resolution methods. But they have found now their way into environmental screening. And data processing tools like mzmine have been used for a long time. So it's just a natural shift also for the environmental people to look at what other fields have already applied successfully. And you can simply translate all these workflows using different spectral libraries, using maybe a few different parameters to accommodate for PFAS molecules compared to regular metabolites.
I touched on ASMS last week. One of your posters focused on deep learning molecular networking for MS dereplication. LC Tim's MS isn't something we've approached on the podcast, so I'd love for you to talk about that and then the poster as a whole.
Yeah, so LC-TIMS MS data obviously has one more dimension, and the deep learning here really touches on how we leverage dreams and MS2 Deep Score for finding even more or being able to identify more molecules than before. So we currently leverage these AI models as a tool to compare spectral similarity of MS2 spectra. Bringing back in the TIMS, so this data was acquired on a TIMS Metabo instrument with the passive acquisition mode. So there we have resolved or mobility resolved MS2 spectra, which is pretty cool because they are quite clean. Even if you have, if you have chromatographic overlap of isobars, that technique may help to create cleaner spectra. And it's also a very fast technique. So you get lots of spectra, again, bringing in the need for automatic data processing. Yeah, and these molecular networking approaches, they really help to cluster molecules of the same group or class or that are actually structurally related. And using available spectral libraries to annotate usually yields in, depending on your libraries and the sample, in maybe 5 or 10%. But if you have a cluster with, let's say, 100 features and 5 of them are annotated, you can then use this as a starting point and go through these networks and look at the single different modifications. And the classic approach here is an algorithm called modified cosine similarity, where we basically shift the spectrum and then do cosine matching. And this works really well for single modifications. But what we see with the deep learning, with DREAMS and with MS2 Deep Score, there we cluster also molecules that are structurally very similar, but yield very different MS2 spectra. So classic modified cosine similarity would never cluster these together. But if we then look at these DREAMS and MS2 Deep Score clusters, we suddenly see, okay, these molecules, they really look very similar, only maybe one functional group is different, but that functional group had a really big impact on the MS2 spectrum. But yet, with the deep learning, we somehow managed to cluster this.
A significant part of the audience for the Concentrating on Chromatography podcast is undergraduates or early career chemists, people just learning about some of these topics in separation science and analytical science. One thing I definitely wanted to touch on was your tiered model for subscriptions, where you have commercial subscriptions, obviously, but you also have free community licenses for academia. For someone who's really excited about the platform and encouraged by this interview, could you provide some background on that? And how do you balance supporting that open source scientific community while also needing to grow a profitable, sustainable business?
Yeah. So we decided to introduce this mzmine academic program where we give out licenses to our software to universities free of charge, to students, to teachers, and scientists working in these labs. And on the other side, we have different tiers for enterprises, depending on team size, the amount of support they need. We make all the technology available to academics. And also, we still publish most of our code open source, especially the processing modules, because we really don't want a black box characteristic. So we really want that people can understand how our algorithms work and why the results look like they do. And this is really, really important to us that we also keep it that way.
And that's been a recurring topic on the podcast as well, especially with some of the instrumentation manufacturers, where there's certainly a push to make science as easy and as quick as possible. But I know in academia, there's also a concern among professors and educators that the analyzers are turning into somewhat of a black box where understanding the science behind what's really going on can sometimes be lost because we've simplified or automated so much of the process. But suppose on the software side, you're actually providing a tool for somebody to grow their understanding and be able to derive more value from the data. Is that an accurate picture?
I mean, that must be the goal to make it easier, but don't oversimplify and try to be transparent with the results. There are always discussions about IDs. How many peptides, how many proteins, how many small molecules can I annotate? And we always try to position ourselves here as open and transparent. Like, okay, this is what you get, but here's all the raw data. Have a look yourself. Here's the dashboard, so you can review it all on your own. And this is also something I'm personally very proud of with the mzmine software, which is also a big difference to most other tools, other processing tools. All our modules, they are really close to the raw data. So what that means is if I have a processing result, a final compound or feature table, with the click of a button, I can always go back into the raw file and inspect an EIC, a spectrum, whatever I want, without needing to open another tool again.
And I always love to hear any insights you could provide on where this is headed. So let's say 5 or so years from now, what does a typical MS data analysis workflow look like for a lab running mzio software?
I think 5 years is quite a long time, especially with the ability that AI gives us, especially in terms of software development. It's a crazy accelerator at this point in time. I think we will move further towards automation. So we have also tools in the pipeline that will help to optimize mzmine processing. Again, for us, this is always the balance to keep all the parameters adjustable for the experts, but also lowering entrance barrier for newcomers. But with AI, here you can actually try to optimize something automatically quite easily. And I think we will move towards something maybe on the acquisition device processing automatically. So I don't see us in 5 years still copying raw data, terabytes on hard disks, or loading it up first to the internet and getting it back. This is actually becoming, data transfer is actually becoming one of the most time consuming parts. It sometimes takes longer than the processing itself. So making our software, let's say AI agent ready, I can see a lot of stuff happening right on the acquisition machine automatically, and then you can adjust later. But getting instant feedback on your measurement and directly see results, maybe even during a sequence, and maybe also feeding them back towards the acquisition. Maybe stop something if something doesn't look right, but maybe also optimize, maybe a DDA method based on already analyzed results.
That makes a lot of sense and not something I had ever thought about, but I could certainly see how that's very time consuming component. Ansgar, thanks so much for introducing your platform and MZIO to the podcast audience. I greatly appreciate it.
Thanks a lot, David, for the invitation. It was very good fun to talk to you.